Oh, this right here, this is my Super Bowl. Mid-autumn is the best time of the year. Not because of dirty chai or blazers or watching the Phillies implode during the postseason or whatever, but because we get this, the sudden global shift in leaf colors from green to yellow to red and beyond. Maybe that seems counterintuitive, but this right here is the single largest and most dramatic display of pure biochemistry that we get in this life on Earth. And honestly, I used to really hate autumn. It's this moment where the joy and glory of green chlorophyll summer suddenly fades to brown as every deciduous tree in the northern hemisphere sheds their leaves and fades to desolation and death ahead of a long, cold winter. But that was mainly due to how I was taught about why trees drop their leaves like this. Most of us learn that the green pigment in leaves, chlorophyll, suddenly fades, revealing other pigments that are yellow, orange, and red underneath. And that barely scratches the surface of what's going on here. In reality, this process, called leaf senescence, is a dramatic race against time, where trees sense the coming winter and work overtime to recycle, repackage, and store all their photosynthetic equipment ahead of a long winter. Ask any basic questions about leaf senescence, like where does the chlorophyll even go? And you'll get incredible insight into all sorts of huge issues and trade-offs life has to deal with to even be alive at all. But be careful, push deep enough into the reasons why leaves burn gold and then red during deep autumn, and you'll also stumble into the ravenous monster hiding at the very heart of photosynthesis. But don't let that stop you. Leaf senescence is one of those processes that will give you so much more respect for the amount of work it takes to even be alive at all. So let's explore all the reasons why leaves fall. You won't want to miss this. Let's get into it. And hey, welcome back to Clockwork, a show about biochemistry. This episode is a quick add-on to my long-running series on photosynthesis. Check out the rest of the playlist if you want to brush up on the basics of how plants get their energy in the first place, and how that has quite literally built the entire world around you. Make sure you subscribe to this channel and check us out at watchclockwork.com to learn even more. But now let's break down this autumn leaf situation. Now, leaf senescence can get pretty complicated, so we're going to break this down into three main parts to make it manageable based on color. We'll start in the bright green chlorophyll-rich world of late summer, and then we'll unravel the complex chemistry that causes trees to burn gold and yellow during the brief reign of carotenoid world. And then yeah, things will take a turn for the dramatic when suddenly some leaves experience emergency conditions so dire that they're forced to turn red using anthocyanins. How on earth could photosynthesis turn out to be dangerous? Before that, let me just hit you with the elevator pitch of why this goes down. Basically, trees have to invest a lot into their leaves during the year in order to turn sunlight, air, and water into food. Leaves have to be chock full of pigments, minerals, proteins, and other complex bits of photosynthetic machinery in order to safely capture sunlight. All this machinery is expensive to build, and it would be super dumb if our trees just abandoned everything and dropped their leaves with all that fancy equipment still inside. So, senescence is a coordinated cellular process where our leaf cells will plan to shut down. In so doing, they will blow up and recycle just about everything they used to photosynthesize. Effectively, all of the nutrients and minerals plants use to photosynthesize will get broken down and sent treeward across September, October, and November before these leaves drop. Sound cool? Awesome. Let's explore the actual details now. Starting with a quick primer. Why do deciduous trees drop their leaves at all? And to really oversimplify here, at their most basic level, trees are giant straws. You have a heart to pump blood throughout your body, but wood and cellulose aren't exactly flexible enough to make that same action happen for trees, so instead, trees evaporate water out of their leaves, and that creates a capillary effect that can pull water out of the ground, bringing water and minerals up through the roots of a tree to the rest of the plant. It's a great passive strategy, honestly, especially since trees are super tall and have a lot of leafy surface area to move a lot of water and capture a ton of sunlight throughout the summer. But if the groundwater these roots are pulling up were to freeze in the winter, our tree is basically going to suffocate. And if that weren't scary enough, the cold temperatures in winter are due to there being less sunlight available to plants thanks to shorter and shorter days. So it really isn't worth the risk of trying to photosynthesize at all during these months. Therefore, smart plants evolved senescence to make sure that they don't end up wasting all the hard work they do during the summer months. Because, and I alluded to this before, photosynthesis is pretty expensive. Trees spend most of the year effectively in debt to themselves. It takes a lot of carbon and energy and protein to make a leaf, and all the photosystems that power photosynthesis. Recycling all of that is a really smart strategy since trees can safely store all those nutrients behind the shield of their bark. So when trees detect that winter is around the corner, that's when senescence is going to kick off. But wait, the signal? Hey, what signal? How do trees know winter is coming? Let's do a quick side quest exploring the science here. This will be a quick one, I promise. There's actually, potentially, a lot of different triggers that kick off the senescence program, but let's focus on the big two. First, trees can sense something called the photoperiod of a given day. Basically, the photoperiod is the amount of strong sunlight that a tree is exposed to within a single 24-hour time frame. During the summer, trees in higher latitudes get exposed to way longer days. More sunlight, more photosynthetic energy production. Nice. In the winter, there are shorter days, which is not so good for photosynthesis. So, when enough shorter sunlight days have been detected by our tree, that's their main trigger for turning off the photosynthesis factory and tucking in for the winter. But second, there's actually been a lot of cool science recently that's pointed to something called source sink dynamics as another, maybe even more important trigger for autumn leaf colors. Basically, while a tree spends most of the year trying to recoup the carbon investment it made in building all its flowers and leaves and new growth, eventually, all of a tree's carbon storage tanks get full. A fully mature leaf is a carbon source. It's donating carbon to the rest of the plant, while storage areas are carbon sinks, taking up all of that carbon. Once all the carbon tanks signal that they're full, it's time to kick off the senescence program, basically. A tree will literally say, hey man, I'm full, time to go to sleep for the winter time, and conk out. Nice. And those two barely scratch the surface of just how much trees and other plants can sense. If you want to explore more into the science of plant intelligence and discover just how much awareness plants have, I can't recommend The Light Eaters by Zoe Schlanger enough. Zoe is one of the absolute goats of science writing, and this is one of the strongest science books of the year. This one gets 5 clocks out of 5, but I wish it had more Arabidopsis. Get ready for more recommendations like this. We're seeing a lot of really great life sciences and chemistry writing these days. But anyway, regardless of what the most important signal here is, senescence is a highly coordinated and complex process of cellular recycling. We're finally here at the main event, so let's uncover the basics of what goes down during the fall here. This program centers around chlorophyll, the main pigment that captures sunlight for plants. Throughout autumn, we're just watching this chlorophyll get broken down. And with that, we're finally ready for the main event. Keep this chart in mind because it'll be one of the main reasons we're seeing big color changes moving forward. But with the signal flipped, our fall is inevitable. Welcome to Phase 1, the opening salvo of cellular degradation. So what breaks down here first? So here's the short story. Basically, the moment the senescence program kicks off, literally everything starts breaking down all at once in these leaf cells. So let me give you a quick lay of the land here first. Leaves may be very thin, but they're still several cells thick. If we drill down a layer here and look at this from the side, you see that the main event is going to happen in this layer called the mesophyll cells. That's kind of where all of the chloroplasts are, and the chloroplasts are the main event of leaf senescence. Inside these mesophyll cells, you'll mostly see chloroplasts, you also see the nucleus of the cell, and this big old vacuole, which is like a bag of water that's slightly more acidic than the surrounding parts of the cell. We're going to get back to our vacuole in a little bit though. So let's zoom into one of our chloroplasts and see where the main event of senescence happens. If you look inside the chloroplast, it contains even more membrane-bound structures made in these little pancake stacks. These are thylakoids, and those stacks are called grana, and the very first thing that really starts happening is that these thylakoid membranes are going to start blowing up and breaking down, because our beautiful chloroplast here is making the wild, awkward, and goofy transition into being something called a gerontoplast, a geriatric chloroplast. And like, what a name, right? Botanists and molecular biologists can be so mean when they want to be. And to start this blow-up process, plants use enzymes like phospholipase A to break down the individual bricks of this membrane. But before this thylakoid totally disintegrates, let's check in on the main event of senescence. If you remember the rest of our photosynthesis series, this thylakoid membrane is where all the magic happens inside the chloroplasts. It's therefore where the main event of senescence takes place. Let's zoom in and see how all this goes down. We're going to visit the various photosystems that power photosynthesis. All of the little coils are proteins, and the green and yellow crystals in the outside are the antennas that are actually absorbing light and passing them on to the proteins. More on that in a second. We need to break down all these proteins, but more importantly, we really need to recycle all this chlorophyll. And since photosystem 2 is my favorite protein in the whole world, let's zero in on them to tell this part of the story. As senescence kicks into high gear, this leaf will build a whole posse of enzymes that end up crowding around these light harvesting complexes. They're here to break down all this chlorophyll for recycling. Ashes to ashes, catabolites to catabolites, folks. It's time to get our nitrogen investment back. Chlorophyll is a bit of a doozy, so it takes a lot of steps to break down into something safe for storage. We're going to bash this center magnesium out, cut off the tail of chlorophyll here, and then over the course of several other steps, rearrange this ring a little bit before this former antenna is now safe to be transferred into the plant cell's big ol' vacuole. In here, things are acidic enough where this once proud chlorophyll antenna can get processed down to something safe for storage. Nice. And while we wrap this up, here's a really interesting bit of botany. Recently, some research has determined that once these chlorophyll catabolites enter the vacuole, they actually stay there for a bit. They'll eventually make their way out and into long-term storage inside the main tree body, but the real rush here is to get chlorophyll broken down and out of the chloroplast, as opposed to just recycling it immediately. Why would that be the focus? Huh. I guess that's just foreshadowing for now. Now, there's so much chlorophyll in these plants that this process happens gradually throughout all of autumn, but eventually enough chlorophyll has been recycled that we can officially reach stage 2 of autumn colors. We're going from green to gold, folks. Chlorophyll is no longer the top dog. Welcome to carotenoid world. So as chlorophyll leaves our example photosystem 2 during this process, things are looking a little shreddy right now, honestly. We'll talk about exactly what's going on with these proteins in a bit, but let's first meet our leaf's brief and beautiful main character, the carotenoids that are left over once chlorophyll starts really getting broken down. This is the supporting cast of photosynthesis. Pigments like neoxanthin, lutein, and the ever-popular beta-carotene here help chlorophyll absorb light and carry out other helpful side jobs within the photosynthetic machinery. And if beta-carotene looks familiar at all to you, well, all you gotta do is chop it up real quick, basically break it in half, and look, you've got vitamin A right here, which is just one quick reaction away from retinal. The same retinal that's absorbing the light being beamed into your eyeballs by this video. If you want to hear more about that, check out the video right before this one in our upload queue. And you know what, quick sidebar here. It's so funny to see a tangential form of retinal end up being a mere support for chlorophyll. You know what, retinal, you really got the short end of the stick here. Everyone knows you were probably the OG engine of photosynthesis before chlorophyll burst onto the scene, rusted 99% of the biosphere to death, kicked off a 300 million year ice age, and completely took over the world, making you second fiddle. But hey, it's cool to see you're still finding work where you can, retinal. That's probably a story for another time, though. Once again, I promise one day I'll make a video about the purple earth hypothesis. But anyway, yeah, carotenoids help out photosynthesis in a lot of ways. Mainly their yellow color comes from their ability to absorb a wider variety of light, allowing them to pass that energy on to chlorophyll. They can also help out when chlorophyll gets overexcited and absorbs too much sunlight, dissipating that extra energy as heat before chlorophyll can hulk out and blow up the photosystem. We're going to need to recycle these carotenoids too, and the program for carotenoid degeneration actually kicks off at the same time that chlorophyll breakdown does, but leaf cells invest a lot more energy into breaking down chlorophyll as quickly as possible for reasons that we'll explore in a second. The difference in urgency leads to the short, golden reign of the carotenoids. When a leaf is fully yellow like this, roughly 75% of its chlorophyll has been recycled into catabolites, while only 50% of the carotenoids have suffered the same fate. This leaf may be yellow, but it's far from done. Remarkably, this leaf is still photosynthesizing with the few scraps of chlorophyll it has left, at least until literally right now, as the first true cold spats hit in October. Then things start getting serious. All of this recycling can actually be very hazardous for our leaves and our trees in general, and throwing even a mild chill into the mix is going to be really scary given how vulnerable our leaf is. We haven't packed up nearly enough material here, so now it's time for some emergency countermeasures. The anthocyanins are now going to take over. But before we do that, let's hear from this video's sponsor, Brilliant. Today's video is sponsored by Brilliant, where you learn by doing. This is an incredible educational service that helps build foundational skills for you from the ground up. I love the interactive lessons over at Brilliant, and I think they're one of the best resources for learning some of the really complex concepts you have to deal with in STEM. Personally, for my unique learning style, the harder topics in math and physics were always extraordinarily difficult for me to tackle in a traditional academic setting, and so for me, Brilliant has been a breath of of fresh air, helping me shore up those foundations. Each lesson is filled with hands-on problem solving that let you actually play with concepts, a method proven to be 6 times more effective than watching lecture videos or suffering through journal clubs. Plus, all content on Brilliant is crafted by an award-winning team of teachers, researchers, and professionals from MIT, Caltech, Duke, Microsoft, Google, and so many more top-tier places. So while you're establishing a real basis of real knowledge on specific topics, it also helps you work those cognitive processes to help you become a better thinker. More importantly, Brilliant is an incremental platform, which will help you develop a powerful daily learning habit. You're not going to learn data science or calculus in a single day, but you can incrementally chip away at the foundations of these problems until you can wrestle with the more advanced concepts. If you want to try everything Brilliant has to offer for free for a full 30 days, visit brilliant.org/clockwork or scan the QR code on screen. Or you can click on the link in the description. You'll also get 20% off an annual premium subscription. Give them a shot, you genuinely won't be disappointed. Let's go ahead and get back into the video at hand. And hey, welcome back. It's time for part 3, Red October. Let's go ahead and see why leaves deploy these anthocyanins. When the weather finally starts cooling off, that's when things get really serious for our leaves. And that's why, usually quite suddenly, some tree species flash from yellow to red, or even sometimes from green to red. The transition from green to yellow makes sense. I mean, we're turning through chlorophyll and revealing the carotenoid supporting cast underneath. Is the same thing like happening with this red color? Are these anthocyanins like beta-carotene supporting cast? Not at all, y'all. Turning red like this takes energy, actually. Once that first cold spat hits, our leaf knows it is in danger, and what you're seeing is a last-ditch emergency countermeasure to keep things safe while the final nutrient packages are getting pumped out of the leaf. The red color is caused by the synthesis and accumulation of these gorgeous pigments, the anthocyanins. These beefy units are what make tomatoes red and grapes purple. But, what exactly are they keeping our leaf safe from? This is why you stick towards the latter part of the video, folks, because even in a regular old video about leaves or whatever, there can be a massive plot twist, and that's where we are right now. Because there is a fundamental fact I have completely glossed over here. And here it is. Plants throw a wall of anthocyanins up at the last minute because, frankly, chlorophyll is dangerous. No, for real. Everyone's favorite green pigment is secretly a monster. The whole process of leaf senescence is actually very high risk for our leaves, but the moment it gets colder, that danger skyrockets. Here's the thing about chlorophyll. It has one job. It absorbs photons and turns that energy from photons into like a little electron laser beam. That's cool and very useful when chlorophyll is safely contained in the architecture of the photosystems, but now we're running into a big problem more and more. Inside these cells, chlorophyll is on the loose here, folks. Remember, most of our big recycling project is happening in parallel, which means the protein architecture of these photosystems is getting gradually chipped away. That means that even though our leaf cell is working very hard to break down all this chlorophyll as fast as possible, odds are a bunch of it will end up free-floating in the interior fluid of these chloroplasts and even in the cytoplasm of our broader leaf cell. And this is very bad. Chlorophyll is just an antenna. It doesn't know where it is, it only knows that it can do neat stuff with photons. So when free-floating chlorophyll gets inevitably hit with a photon, it will still absorb and try to transfer that energy. The only thing nearby, though, is going to be the water molecules chlorophyll is free-floating through. So therefore, chlorophyll is going to turn into a molecular death ray and start splitting water molecules all willy-nilly. Inside the protein of a photosystem, that's great, we can use this laser beam to make food. Outside of a photosystem, all we're doing here is zapping water molecules and creating reactive oxygen species. You know, free radicals. These are dangerous oxygen variants that have been energized to a violent extent. They roam through solutions in cytoplasm with a ravenous hunger for the blood of loose electrons, and they don't care what they have to tear into in order to get to those electrons. And if you think this sounds kinda bad, wow, you're so right! Here's a non-comprehensive list of things that produce reactive free oxygen species, like inside the average living thing. First up, you got UV radiation, nuclear radiation, severe cellular stress, overpowered acids and bases, and basically, you know, all the worst things. Let me really drive this home. If you get exposed to a whole bunch of nuclear radiation and don't, like, get instantly incinerated or whatever, it's not actually the radiation itself that's gonna kill you. Instead, the cascade of nuclear radiation washes over your cells, producing whole armies of these reactive oxygen species. And it's those monsters that will gradually liquefy your insides, obliterate your DNA, and do all the damage everyone's talking about. So yeah, if we just blow up this photosystem and leave all this chlorophyll lying around, congratulations! Your chloroplast is now a big old bag of randomly firing death rays. Awesome. This is something called phototoxicity, and it's actually a problem plants have to deal with year-round. Expose a photosystem to too much sunlight, and chlorophyll can flip out and cause severe damage. That's one of the main reasons carotenoids are here at all. They take away excess energy when things get too heated before chlorophyll can enter cancer mode, basically. But back to our actual moment here in leaf senescence, as things cool off, photosynthetic efficiency also drops. This can increase the likelihood of phototoxicity in senescing leaves. So it really is time to pump the brakes on the whole photosynthesis thing here in the first place, which finally brings us back to these anthocyanins. These fellas are red because they reflect red light while absorbing high-energy green wavelengths. So anthocyanins are built up to keep things from getting out of hand. There's a lot of debate about how anthocyanins work and what exactly their real benefit is. But the headline here is that our leaf cell will start ramping up anthocyanin production as a photoprotectant. By accumulating these red pigments in the vacuole, leaves are creating a filter that blocks higher energy light from hitting loose chlorophyll inside our degrading chloroplasts. There's also research that shows that anthocyanins have antioxidant properties, so they could be there entirely just to absorb the brunt of the damage being caused by reactive oxygen species. Anthocyanins become a protective shield for the leaf in the last few days of senescence. Remember, every drop of nutrition counts in tree world, especially when winter is about to hit. Anthocyanin production starts pretty early during the fall, but it kicks into high gear, usually after the first real bad cold spat. That's a strong enough signal for our leaves that the party is definitely about to end. During this uptick in anthocyanin production, we've churned through the last of our chlorophyll. With almost nothing left to give, our leaf cell now is entering into the last phase of senescence. And so all the degradation and recycling we've talked about so far is just going to ramp up as this fire sale accelerates. Which means now our leaves have entered the terminal spiral. We're about to really pack things up and drop this structure here. But it's not all bad, right? This last-ditch shielding by anthocyanins has bought a few critical days where our leaves can use what energy is left to finish off the leaf evacuation. Which brings us to that final phase. Our chloroplast is going to complete the transformation into a gerontoplast and stop being the primary energy source for our cell. At this moment, the mitochondria in our leaf cell kick into overdrive and become the primary energy providers here. In these final days, plants briefly get all their energy the same way you and I do, also via some very convenient and occasional self-cannibalism. The thylakoid membranes in this chloroplasts are going to finish imploding, and our once proud pancake stack is going to flatten into one long, floppy, sad little crepe. The phospholipids here are going to continue getting broken down, and some may even end up being used by mitochondria to make energy via ketogenic pathways. Meanwhile, nutrient evacuation is in full swing. Welcome to the fire sale. Everything must go. All those chlorophyll catabolites will get pumped out, along with most of the iron, phosphorus, and nitrogen that was originally in this leaf. Even the leaf cell's DNA gets broken down and shipped out to a certain extent. Back at photosystem 2, proteases are continuing to chip away at this architecture until the entire structure is little chunks a handful of amino acids long. Or proteins get tagged with ubiquitin and then blenderized by this incredible structure, the proteasome. And no, really, this fella is basically the molecular equivalent of a blender. Big proteins come in, little bits of amino acids come out ready to be transformed into new proteins. But this structure is just something else. Again, we're going to have to cover it in more depth in another video, someday. This amino acid surge will get streamed out of our leaf and shoved down into the phloem water transport system inside the main body of the tree. There, all these protein bits will get resynthesized into something called bark storage proteins. Proteins that store energy in the bark of a tree. I love naming conventions. This is a stable enough place to hide your energy while things hit the deep freeze during winter. For now, we're approaching the final moments here as the likelihood of a first frost starts to skyrocket. At this stage, the central tree is even starting to reclaim the water from this leaf. The internal cytoplasm of our mesophyll cells starts to turn thick and almost gelatinous as it pulls away from the cell wall. And you know what? That's about all our hero can give. After a full growing season of service, it's time to cut bait and let this leaf fall. A final act here is the erection of a barrier membrane between our leaf and the branch that held it. All we need is an autumn gust of sufficient strength and, you know what? We're out of here. This leaf will still briefly cling to life though. I mean, what else was it going to do? As energy reserves run out, all the energetic pigments inside this leaf will either get cannibalized or oxidize. At the end, the final color of our leaf is this desiccated brown, caused by all the once energetic macromolecules literally rusting over. But it's not all in vain though, right? Because from the ashes of sacrifice will spring the hope for new life. Our tree has saved up an incredible surplus of energy that it will use to burst forth the moment things get warm and bright enough come March. For now, it's just a good time to rest though. So let's go ahead and just wrap this thing up. I won't lie to you, right? Autumn used to be my least favorite season. After the warmth and light and life of chlorophyll summer, the slow slide to carotenoid world and beyond was just like a major bummer. Watching that gold gradually fall to brown, I always felt like I was surrounded by so much death. A giant cruel joke right before the desolation and darkness of a long winter. But like so many other times, context can be a source of joy in moments like this. These leaves aren't dying. The chlorophyll isn't just going away. Wintering trees aren't even dead. Instead, autumn is this dramatic, astonishing race against time where our deciduous neighbors squeeze every last possible drop they can out of the marrow of life before turning dormant and getting some well-deserved rest through the winter. Life is about those small, incremental triumphs you can make over chaos and death. And these autumn colors are a brief, beautiful flash that will show you just how much work it is to be alive at all. Even if chlorophyll will absolutely atomize you who was ever given the chance. But that's a good enough place to end it. I really appreciate your time. Thank you so much for listening. And hey, thank you so much for watching all the way to the end. I can't tell you how much that means to me. If you like this video, please consider subscribing to this channel, and even more so, I'd love it if you checked us out over at Patreon. Otherwise, another easy way you can support the channel is going over to watchclockwork.com and signing up for our free newsletter, where eventually I will get back into doing little mini Clockwork episodes that just live in your inbox. It's a great way to sort of explore the cutting edge of what's happening in biochemistry. Again, I'd also love it if you consider checking out our sponsor over at brilliant.org/clockwork. Once again, you can get a 30-day free trial from there, as well as 20% off a premium annual subscription. And I legitimately can't recommend the service enough. Otherwise, I really appreciate your time. As always, I like to leave you with peace, love, and cytochrome B6F. Everyone be well. Thank you so much.